Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover other marine animals with fascinating characteristics. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. The mantis shrimp, Odontodactylus ciliaris*, strikes with the acceleration of a .22 caliber bullet and sees with 16 types of photoreceptors. Humans have 3. The archerfish, Toxotes jaculatrix*, spits jets of water with pinpoint accuracy to knock insects off branches up to 3 meters above the surface, instinctively correcting for the optical distortion caused by refraction between water and air. The blue-ringed octopus, Capiloclina *sp, fits in the palm of your hand and carries enough venom to kill 26 adults, with no antidote available. The trumpetfish, Elastomus maculatus *, uses larger fish as a living shield to sneak up on prey without being noticed, the only documented case of an animal using another as a form of concealment. The lantern shark, Etmopterus spinax *, uses bioluminescence to erase its own shadow with belly light to become invisible. The mimic octopus, Thermoctopus mimicus *, impersonates in real time up to 16 different species, flounder, lionfish, sea snake, choosing its disguise based on the predator at hand. The pufferfish, Tetraodontidae, concentrates tetrodotoxin, 1,200 times more toxic than cyanide, in its own organs. The spiny lobster, Panilaris argus *, migrates single file across the ocean floor, each individual touching the one ahead with its antennae, cutting hydrodynamic drag by 65%. Cuvier's beaked whale, Xiphias cavarostris *, dives to 2,992 meters and stays submerged for 3 hours and 42 minutes, both all-time records among mammals. And the snipe eel, Nemicthes scolopaceus *, is born as a transparent leaf-shaped larva, the leptocephalus, that actually shrinks before transforming into an adult. Imagine punching through a glass wall with your bare fists. Now imagine doing it with appendages smaller than a finger. That's the starting point for understanding what lives in the oceans. Creatures that exist as living proof that physics, chemistry, and biology can take on forms radically different from anything we know on land. 10 species in particular carry properties that, on their own, would already be extraordinary. Together, they form a catalog of strategies nature spent millions of years refining. The mantis shrimp leads this list not just for its punch, but for the sensory system that backs it up. With 16 types of photoreceptors spread across eyes that move independently of each other, this crustacean doesn't see the world in greater richness than humans. It sees it in a completely different way. Its vision works like a rapid scan and classify system, identifying patterns before any detailed brain processing even kicks in. The archerfish solves a physics problem that would stump engineers without a calculator. When light moves from water to air, it changes direction, an effect called refraction. A target that appears to be at a certain position above the surface is actually displaced at an angle. The archerfish corrects for that shift instinctively, adjusting the trajectory of its shot to hit the target's actual position, not the apparent one. It lands hits on targets up to 3 meters high. The blue-ringed octopus carries tetrodotoxin in its salivary glands, the same neurotoxin found in pufferfish. At rest, it shows a dull, muted coloration. When threatened, iridescent blue rings pulse across its body as a warning. That visual signal doesn't dial down the danger. The toxin paralyzes the respiratory muscles, causes death by asphyxiation, and has no known antidote. An animal that fits in an adult's hand carries enough venom to kill dozens of people. The trumpetfish developed a camouflage strategy that has nothing to do with color or texture. Getting close to alert prey requires cover. The trumpetfish uses the body of larger fish as a blind. It swims alongside harmless species, tucking itself laterally against them, invisible from the perspective of whatever prey lies ahead. It's the only scientifically documented case of an animal using another individual of a different species as an active concealment tool. The lantern shark solves the opposite visibility problem. In mid-depth waters, predators look upward and spot prey by the contrast of their silhouette against the diffuse light filtering down from above. The lantern shark has photophores, bioluminescent organs, distributed across its belly. These organs emit light that matches the ambient brightness coming from above, wiping out the contrast and making the silhouette invisible to anything looking up from below. The mimic octopus doesn't just change color and texture like other cephalopods. It actively reshapes its posture, body form, and behavior to impersonate completely different species, from flat-bodied flounders to sea snakes with venomous color patterns. The choice of imitation doesn't seem random. Different predators trigger different impressions, suggesting the animal is actually sizing up the threat and picking the most effective response. The spiny lobster carries out seasonal migrations in continuous single-file lines along the ocean floor, each animal keeping its antennae in contact with the one ahead. Flow tunnel tests showed that this formation cuts hydrodynamic drag by up to 65% for individuals in the middle of the line, a mechanism similar to what cyclists use in racing packs and aircraft apply in flight formations. Cuvier's beaked whale holds 2 simultaneous mammal world records. The deepest dive ever recorded, at 2,992 meters, and the longest continuous submersion, at 3 hours and 42 minutes. To pull that off, it compresses its lungs nearly to collapse on the way down, stores oxygen in muscle myoglobin, and dramatically slows its heart rate. Its heart drops to fewer than 10 beats per minute during the dive. The snipe eel goes through an unusual metamorphosis. Its larval stage, the leptocephalus, is a glassy, leaf-shaped, translucent structure that drifts through ocean currents. At a certain point, that structure physically shrinks before reorganizing itself into an adult. Losing mass and length during development runs counter to the expected pattern of continuous growth, making this animal one of the few documented examples of shrinkage as a metamorphic transition stage. The mantis shrimp can strike with its appendages at speeds of 23 meters per second, 50 times faster than the blink of an eye, roughly the same velocity as a .22 caliber round, and with a force about 100 times its own body weight, making it the most powerful self-propelled strike of any animal. The dactyl slowly builds up energy using its muscles, locks the mechanism, then releases the latch. The punch is functionally ballistic. The mechanism is a saddle-shaped spring made of mineralized cuticle. The release is so violent it creates cavitation bubbles in the water, vacuum bubbles that collapse with an audible snap, generating a second shockwave that hits the prey even when the direct strike misses. As for the eyes, some species have at least 16 types of photoreceptors, split into 4 classes. 12 for color analysis across different wavelengths, including 6 sensitive to ultraviolet light, and 4 for analyzing polarized light. Despite that, behavioral studies show the shrimp is actually worse than humans at telling apart closely similar colors. The visual system appears to work more like a fast identification scanner than a rich color palette. No muscle, on its own, can release energy fast enough to generate that kind of speed. Physics sets a hard limit. Muscles contract more slowly than the time window required for a ballistic strike. The mantis shrimp got around that constraint with a biological engineering solution. It builds up energy slowly, then releases it in a fraction of a millisecond, decoupling the loading time from the firing time. The key is a special saddle-shaped joint that stores elastic energy. This structure, built from mineralized cuticle, the same hardened material that makes up the animal's exoskeleton, deforms during loading like a compressed spring. When the latch releases, the stored energy converts instantly into movement of the dactyl, the terminal appendage that delivers the impact. The whole process happens too fast to follow with the naked eye. The impact creates cavitation, a physical phenomenon where pressure drops so low that water momentarily boils, forming vapor bubbles. When those bubbles collapse, they release heat and light in a tiny flash, along with a second impact wave. Think of it as a punch that hits twice. First the direct impact, then the shock wave from the collapsing bubbles. Prey can be incapacitated or killed by that second wave, even if the appendage never made direct contact. The mineral density in these legs is so high that they can withstand thousands of strikes without suffering fatigue fractures, giving the individual impressive functional longevity. That represents another engineering problem solved biologically. Materials rigid enough to deliver high-energy impact tend to be brittle. The mantis shrimp's mineralized cuticle combines hardness with an internal structure capable of dissipating stress waves without propagating fractures. The raptorial appendages work like a catapult mechanism that stores elastic energy and releases it almost instantaneously. Similar mechanisms show up in other contexts. Medieval catapults stored energy by slowly turning a counterweight and released it all at once. Bombardier beetles store separate chemicals and combine them in a reaction chamber with explosive results. The principle, decoupling loading from firing, keeps showing up wherever physics puts a ceiling on direct muscle contraction speed. Some species have at least 16 photoreceptors, split into classes. 12 for color analysis across different wavelengths, including 6 sensitive to ultraviolet light, and 4 for analyzing polarized light. For comparison, human eyes have 3 types of cones, sensitive to red, green, and blue, and all the color perception humans know comes from combining those 3 inputs. 16 channels would, hypothetically, represent an immeasurable perceptual richness. The paradox is that the mantis shrimp doesn't use that architecture to discriminate colors with superior precision. Instead of just seeing a richer rainbow, its vision works more like a fast scanner for identifying patterns, prey, rivals, and mates with high efficiency. Think of a camera that, rather than gradually building up an image, classifies the object in fractions of a second by category: prey, predator, mate, territory. The abundance of channels serves speed of classification, not subtlety of discrimination. This system allows visual information to be pre-processed by the eyes rather than the brain, which would otherwise need to be larger to handle the raw data flow, requiring more time and energy. In the human visual system, color processing happens in cortical regions. In the mantis shrimp, part of that processing takes place right at the retina, cutting the time between stimulus and response. For a predator that strikes in fractions of a second, that speed gain can be the difference between eating and losing the prey. The mantis shrimp can move its eyes independently and is the only known animal capable of detecting circularly polarized light. Polarized light vibrates in a specific plane. Circularly polarized light rotates that plane in a spiral. Biological surfaces reflect those patterns in distinct ways, making it possible to identify materials by their polarization signature, not just by color or shape. No other known animal makes that distinction. Modern engineering has already used this crustacean's eye as a model for developing equipment that detects cancer. Since cancerous cells reflect polarized light differently than healthy cells, cameras inspired by the mantis shrimp may help doctors identify tumors at early stages with far greater precision than current methods allow. Marine biology, in this case, translated directly into applied medical technology. The mantis shrimp, Odontodactylus ciliaris*, strikes with the acceleration of a .22 caliber bullet and sees with 16 types of photoreceptors. Humans have 3. The archerfish, Toxotes jaculatrix*, spits jets of water with pinpoint accuracy to knock insects off branches up to 3 meters above the surface, instinctively correcting for the optical distortion caused by refraction between water and air. The blue-ringed octopus, Capiloclina *sp, fits in the palm of your hand and carries enough venom to kill 26 adults, with no antidote available. The trumpetfish, Allostomus maculatus*, uses larger fish as a living shield to sneak up on prey without being noticed, the only documented case of an animal using another as a form of concealment. The lantern shark, Etmopterus spinax*, uses bioluminescence to erase its own shadow with belly light to become invisible. The mimic octopus, Thermoctopus mimicus*, impersonates in real time up to 16 different species: flounder, lionfish, sea snake, choosing its disguise based on the predator at hand. The pufferfish, Tetraodontidae, concentrates tetrodotoxin, 1,200 times more toxic than cyanide, in its own organs. The spiny lobster, Panularis argus*, migrates single file across the ocean floor, each individual touching the one ahead with its antennae, cutting hydrodynamic drag by 65%. Cuvier's beaked whale, Xiphias cavarostris*, dives to 2,992 meters and stays submerged for 3 hours and 42 minutes, both all-time records among mammals. And the snipe eel, Nemicthes scolopaceus*, is born as a transparent leaf-shaped larva, the leptocephalus, that actually shrinks before transforming into an adult. Imagine punching through a glass wall with your bare fists. Now imagine doing it with appendages smaller than a finger. That's the starting point for understanding what lives in the oceans. Creatures that exist as living proof that physics, chemistry, and biology can take on forms radically different from anything we know on land. 10 species in particular carry properties that on their own would already be extraordinary. Together, they form a catalog of strategies nature spent millions of years refining. The mantis shrimp leads this list not just for its punch, but for the sensory system that backs it up. With 16 types of photoreceptors spread across eyes that move independently of each other, this crustacean doesn't see the world in greater richness than humans. It sees it in a completely different way. Its vision works like a rapid scan and classify system, identifying patterns before any detailed brain processing even kicks in. The archerfish solves a physics problem that would stump engineers without a calculator. When light moves from water to air, it changes direction, an effect called refraction. A target that appears to be at a certain position above the surface is actually displaced at an angle. The archerfish corrects for that shift instinctively, adjusting the trajectory of its shot to hit the target's actual position, not the apparent one. It lands hits on targets up to 3 meters high. The blue-ringed octopus carries tetrodotoxin in its salivary glands, the same neurotoxin found in pufferfish. At rest, it shows a dull, muted coloration. When threatened, iridescent blue rings pulse across its body as a warning. That visual signal doesn't dial down the danger. The toxin paralyzes the respiratory muscles, causes death by asphyxiation, and has no known antidote. An animal that fits in an adult's hand carries enough venom to kill dozens of people. The trumpetfish developed a camouflage strategy that has nothing to do with color or texture. Getting close to alert prey requires cover. The trumpetfish uses the body of larger fish as a blind. It swims alongside harmless species, tucking itself laterally against them, invisible from the perspective of whatever prey lies ahead. It's the only scientifically documented case of an animal using another individual of a different species as an active concealment tool. The lantern shark solves the opposite visibility problem. In mid-depth waters, predators look upward and spot prey by the contrast of their silhouette against the diffuse light filtering down from above. The lantern shark has photophores, bioluminescent organs, distributed across its belly. These organs emit light that matches the ambient brightness coming from above, wiping out the contrast and making the silhouette invisible to anything looking up from below. The mimic octopus doesn't just change color and texture like other cephalopods. It actively reshapes its posture, body form, and behavior to impersonate completely different species, from flat-bodied flounders to sea snakes with venomous color patterns. The choice of imitation doesn't seem random. Different predators trigger different impressions, suggesting the animal is actually sizing up the threat and picking the most effective response. The spiny lobster carries out seasonal migrations in continuous single-file lines along the ocean floor, each animal keeping its antennae in contact with the one ahead. Flow tunnel tests showed that this formation cuts hydrodynamic drag by up to 65% for individuals in the middle of the line, a mechanism similar to what cyclists use in racing packs and aircraft apply in flight formations. Cuvier's beaked whale holds 2 simultaneous mammal world records. The deepest dive ever recorded, at 2,992 meters, and the longest continuous submersion, at 3 hours and 42 minutes. To pull that off, it compresses its lungs nearly to collapse on the way down, stores oxygen in muscle myoglobin, and dramatically slows its heart rate. Its heart drops to fewer than 10 beats per minute during the dive. The snipe eel goes through an unusual metamorphosis. Its larval stage, the leptocephalus, is a glassy, leaf-shaped, translucent structure that drifts through ocean currents. At a certain point, that structure physically shrinks before reorganizing itself into an adult. Losing mass and length during development runs counter to the expected pattern of continuous growth, making this animal one of the few documented examples of shrinkage as a metamorphic transition stage. The mantis shrimp can strike with its appendages at speeds of 23 meters per second, 50 times faster than the blink of an eye, roughly the same velocity as a .22 caliber round, and with a force about 100 times its own body weight, making it the most powerful self-propelled strike of any animal. The dactyl slowly builds up energy using its muscles, locks the mechanism, then releases the latch. The punch is functionally ballistic. The mechanism is a saddle-shaped spring made of mineralized cuticle. The release is so violent it creates cavitation bubbles in the water, vacuum bubbles that collapse with an audible snap, generating a second shockwave that hits the prey even when the direct strike misses. As for the eyes, some species have at least 16 types of photoreceptors, split into 4 classes, 12 for color analysis across different wavelengths, including 6 sensitive to ultraviolet light, and 4 for analyzing polarized light. Despite that, behavioral studies show the shrimp is actually worse than humans at telling apart closely similar colors. The visual system appears to work more like a fast identification scanner than a rich color palette. No muscle on its own can release energy fast enough to generate that kind of speed. Physics sets a hard limit. Muscles contract more slowly than the time window required for a ballistic strike. The mantis shrimp got around that constraint with a biological engineering solution. It builds up energy slowly, then releases it in a fraction of a millisecond, decoupling the loading time from the firing time. The key is a special saddle-shaped joint that stores elastic energy. This structure, built from mineralized cuticle, the same hardened material that makes up the animal's exoskeleton, deforms during loading like a compressed spring. When the latch releases, the stored energy converts instantly into movement of the dactyl, the terminal appendage that delivers the impact. The whole process happens too fast to follow with the naked eye. The impact creates cavitation, a physical phenomenon where pressure drops so low that water momentarily boils, forming vapor bubbles. When those bubbles collapse, they release heat and light in a tiny flash, along with a second impact wave. Think of it as a punch that hits twice. First the direct impact, then the shock wave from the collapsing bubbles. Prey can be incapacitated or killed by that second wave, even if the appendage never made direct contact. The mineral density in these legs is so high that they can withstand thousands of strikes without suffering fatigue fractures, giving the individual impressive functional longevity. That represents another engineering problem solved biologically. Materials rigid enough to deliver high-energy impact tend to be brittle. The mantis shrimp's mineralized cuticle combines hardness with an internal structure capable of dissipating stress waves without propagating fractures. The raptorial appendages work like a catapult mechanism that stores elastic energy and releases it almost instantaneously. Similar mechanisms show up in other contexts. Medieval catapults stored energy by slowly turning a counterweight and released it all at once. Bombardier beetles store separate chemicals and combine them in a reaction chamber with explosive results. The principle, decoupling loading from firing, keeps showing up wherever physics puts a ceiling on direct muscle contraction speed. Some species have at least 16 photoreceptors, split into classes. 12 for color analysis across different wavelengths, including 6 sensitive to ultraviolet light and 4 for analyzing polarized light. For comparison, human eyes have 3 types of cones, sensitive to red, green, and blue, and all the color perception humans know comes from combining those 3 inputs. 16 channels would, hypothetically, represent an immeasurable perceptual richness. The paradox is that the mantis shrimp doesn't use that architecture to discriminate colors with superior precision. Instead of just seeing a richer rainbow, its vision works more like a fast scanner for identifying patterns, prey, rivals, and mates with high efficiency. Think of a camera that, rather than gradually building up an image, classifies the object in fractions of a second by category: prey, predator, mate, territory. The abundance of channels serves speed of classification, not subtlety of discrimination. This system allows visual information to be pre-processed by the eyes rather than the brain, which would otherwise need to be larger to handle the raw data flow, requiring more time and energy. In the human visual system, color processing happens in cortical regions. In the mantis shrimp, part of that processing takes place right at the retina, cutting the time between stimulus and response. For a predator that strikes in fractions of a second, that speed gain can be the difference between eating and losing the prey. The mantis shrimp can move its eyes independently and is the only known animal capable of detecting circularly polarized light. Polarized light vibrates in a specific plane. Circularly polarized light rotates that plane in a spiral. Biological surfaces reflect those patterns in distinct ways, making it possible to identify materials by their polarization signature, not just by color or shape. No other known animal makes that distinction. Modern engineering has already used this crustacean's eye as a model for developing equipment that detects cancer. Since cancerous cells reflect polarized light differently than healthy cells, cameras inspired by the mantis shrimp may help doctors identify tumors at early stages with far greater precision than current methods allow. Marine biology, in this case, translated directly into applied medical technology. The mantis shrimp represents, in miniature, proof that evolution and engineering arrive at the same solutions by completely independent paths. Springs, latches, catapults, rapid identification systems, polarization sensors. Every one of those mechanisms has been discovered and rediscovered by human engineers. The crustacean packed all of them into a 16-centimeter body. The question that remains isn't whether this animal is extraordinary. It's what else it knows that we haven't figured out yet. When an object is above the water's surface and the observer's eyes are below it, light traveling between the two mediums bends at the boundary. That's refraction. The target appears to be somewhere it isn't. The archerfish must compensate for the refraction distortion that makes objects look roughly 25% closer and higher than their actual position, adjust for the drop of the projectile over distance, and even predict the trajectory of moving targets. A precise firing angle, adjusted for position and distance, to compensate for an optical effect that the fish's own eyes can't directly correct for. An adult fish almost always hits its target on the first try. They can knock down insects and other prey up to 3 meters above the water's surface. They generally fire at an average angle of about 74 degrees relative to horizontal, but can still aim accurately anywhere between 45 degrees and 110 degrees. Research has shown that the archerfish learns to correct for this distortion through a process of motor adaptation, trial and error, adjusting its shots until it nails the calculation. Imagine trying to hit a ball that isn't where your eyes say it is. That's exactly what the archerfish deals with every time it hunts. The light leaving an insect perched on a branch travels through air until it hits the water's surface, and at that point, it changes direction. The result is that the insect appears to be in a different spot than it actually is, shifted upward at an angle and seemingly closer. Any projectile aimed at that imaginary point would miss completely. The species calculates the refraction of light to hit prey outside the water. The technique involves curling the tongue into a tube and closing the gill covers, creating a continuous jet of water that knocks insects off leaves and branches. This mouth mechanism works like a biological pressure hose, capable of adjusting the force and volume of the jet based on the distance and size of the target. The physics of the problem has 2 separate components. The first is the positional illusion caused by refraction. The insect appears to be where it isn't. The second is the ballistics of the jet itself. Water in a parabolic arc drifts off course the farther it travels. When the fish fires its jets, it modifies the water flow to give its aim greater precision. This indicates the animal isn't just calculating where to aim. It's actively controlling the shape of the projectile in flight. What makes this behavior even more remarkable is that no specialized structure automatically compensates for refraction. The archerfish's eyes experience the same distortion as any other submerged eye. The correction isn't anatomically automatic. It's computed by the central nervous system on every single shot, adjusted for the viewing angle, the estimated distance, and the height of the target. All of that without a single measuring instrument. The archerfish has high precision at distances of 1.2 to 1.5 meters, and occasionally succeeds at distances of up to 3.5 meters. That precision window isn't random. It corresponds to the range where the animal can most reliably estimate the parameters needed for the calculation. As distance increases, the variables multiply and the margin for error grows. Researchers have shown that juveniles miss their first shots and gradually improve their accuracy. This confirms that the correction isn't instinctive from birth. It's learned through trial and error, a gradual calibration process that fine-tunes each individual's internal parameters. The archerfish also has excellent binocular vision, which lets it calculate the exact distance to its prey, sharpening the effectiveness of its shot even further. The average firing angle of 74 degrees relative to horizontal isn't arbitrary. Shooting nearly straight up minimizes the distance the jet has to travel through water before reaching the surface, reducing energy loss from drag. At the same time, it requires the animal to position itself almost directly below the target, which isn't always possible. That's why researchers who study the species say it has calibrated precision and controls even the speed of the jet, which has to be strong enough, but not too strong, to bring the target down. A moving target adds a third variable, trajectory prediction. Insects in flight or walking across leaves change position during the time the jet is in the air. The archerfish has to aim at where the insect will be when the jet arrives, not where it is at the moment of firing. That involves estimating the target's speed and direction, an operation that requires integrating visual information over time. The practical upshot is that college students in optical physics class learn the refraction problem with equations and problems on paper. The archerfish solves the same problem in real time, without any formulas, at every meal. The calculation is identical. The way it gets processed is radically different. Neither one could execute the other's solution. The shadowing behavior has been observed on degraded reefs, suggesting a possible adaptive strategy in response to environmental change. Though that observation refers to the trumpetfish, it points to a universal principle. When the environment changes, the animals that survive are the ones that can recalibrate their behavior. The archerfish, for its part, recalibrates the physics with every shot. The blue-ringed octopus rarely measures more than 22 centimeters from the tip of its mantle to the ends of its arms. At rest, it's a dull beige, nearly invisible among rocks and coral. When threatened, the rings light up in vivid iridescent blue, not as an aesthetic choice, but as a warning signal. The bite is tiny and often painless. Many victims don't realize they've been envenomated until respiratory depression and paralysis begin setting in. The venom is tetrodotoxin, 1,000 times more potent than cyanide. A single blue-ringed octopus carries enough venom to kill 26 humans in minutes. There is no antidote. Death typically comes from respiratory failure, after the diaphragm has been paralyzed. This can happen within minutes. There is no known antidote for TTX, and most care is supportive ventilation to keep the patient breathing until the toxin's effects wear off. The venom isn't produced by the animal itself, but by symbiotic bacteria in its salivary glands. Few animals solve the problem of survival in a way so economically brutal. Too small to intimidate predators by size, too slow to escape efficiently, the blue-ringed octopus concentrated its entire defensive strategy into a single resource: surgical precision venom, announced by a visual signal that's impossible to ignore. Blue-ringed octopuses don't make the tetrodotoxin themselves. Instead, the toxin is produced by symbiotic bacteria that live in their salivary glands, according to the Australian Institute of Marine Science. Tetrodotoxin is found in all of the octopus's tissues, not just in specific venom glands, which makes them among the few animals that are both venomous and poisonous to the touch. That means the danger isn't limited to the bite. Even simple skin contact with the animal can transfer the toxin. The venom prevents nerves from sending signals to muscles by blocking sodium ion channels. This causes rapid muscle weakness and paralysis, including in the respiratory tract, which can lead to respiratory arrest and death. To understand the mechanism, nerve cells need sodium flow to generate electrical impulses. Without that flow, the signal doesn't exist. And without the signal, the muscle receives no command to contract. The diaphragm stops. Breathing ceases. The heart may keep beating for a while, but without ventilation, oxygen in the blood runs out fast. The detail that makes this scenario even more disturbing is the order in which events unfold. The toxin shuts down natural breathing without affecting the victim's consciousness. The person remains awake, able to think and perceive their surroundings, while the muscles become increasingly unresponsive. The paralysis works through the limbs, then the chest, and finally the diaphragm, but leaves consciousness intact until cerebral hypoxia finally extinguishes thought as well. The paralysis hits voluntary muscles, causing death by oxygen deprivation while the victim remains fully conscious. There is no known antidote. but the person can be saved if artificial respiration is started immediately. The treatment is mechanical, replacing for hours the work of the paralyzed diaphragm while the body metabolizes and eliminates the toxin. Artificial ventilation keeps the victim alive. Without it, the window is a matter of minutes. According to the U.S. Centers for Disease Control and Prevention, death can occur anywhere from 20 minutes to 24 hours after exposure. That variation depends on the dose absorbed, the entry point of the toxin, and how quickly the victim receives treatment. Bites in highly vascularized areas deliver the toxin to the central nervous system faster than bites on the extremities. The iridescent blue rings flashing across the octopus's body warn predators of its toxicity. This warning system is called aposematism, coloration that communicates danger rather than concealing it. The logic is straightforward. A predator that learns to associate pulsing blue rings with lethal consequences avoids the animal with no confrontation needed. The signal works like a biological warning label that protects both the predator and the octopus. The amount of tetrodotoxin stored in a single individual can be enough to cause multiple human deaths if administered in the right dose. Concentrating that arsenal in a 22-centimeter body represents a high-yield solution. Minimal mass, maximum deterrent effect. No reasonable predator attacks twice after a first encounter with this species. Studies are investigating how the bacteria are acquired and whether blue-ringed octopuses lose their ability to produce toxins when removed from the wild and placed in a controlled environment like an aquarium. This is a relevant point. If the toxin depends on bacteria obtained in the wild, captive-raised animals may not be lethal in the same way, which makes the relationship between the octopus and its microbiome an essential piece of its survival strategy. The blue-ringed octopus is ultimately a demonstration that size and toxicity are independent variables. An animal that fits comfortably in the palm of a hand carries enough venom to neutralize dozens of much larger organisms. With no antidote, no perceptible warning in the bite, and an intervention window that closes fast. The blue iridescence isn't decoration. It's the final communication before irreversible consequences. A new study provides the first experimental evidence that the trumpetfish, Allostomus maculatus, can hide by swimming close to another fish while hunting and reduce the odds of being spotted by prey. In this shadowing behavior, the long, slender trumpetfish uses a non-threatening species, like a parrotfish, as camouflage to sneak up on dinner. This is the only known example of a non-human animal using another animal as a form of concealment. When the trumpetfish swims close to another fish species, it's either completely hidden from its prey, or it's seen but not recognized as a predator because the shape is different. Researchers found that shadowing behavior was more common on more degraded and less structurally complex coral reefs. The trumpetfish tends to choose shield fish whose coloration is similar to its own, maximizing visual camouflage within the behavioral camouflage. In the vast majority of documented cases, camouflage is a property of the body, color patterns, textures that mimic substrate, shapes that blend into the environment. The trumpetfish threw out that logic entirely. Its body is long, narrow, and impossible to disguise among complex corals. The solution it landed on wasn't to change its appearance. It was to change the context in which that appearance is perceived. When a trumpetfish swims near another fish species, it either stays completely hidden from its prey or is spotted, but not recognized as a predator because the shape is different, explained Dr. Sam Machette, who led the study. The distinction between the two possibilities matters. In some cases, the trumpetfish physically hides behind its shield, completely out of sight. In others, it's visible, but integrated into a two-fish configuration where the combined silhouette doesn't match the pattern that prey recognizes as a threat. Diverse regularly report seeing the trumpetfish swimming alongside a larger, typically less threatening fish, like a parrotfish or surgeonfish, in a practice known as shadowing. The phenomenon had been observed for years, but only as a field curiosity, experimentally confirming that the behavior actually reduces detection by prey-required controlled methodology, which the Cambridge team carried out using 3D printed models tested on the reef itself. Since they're very poor swimmers, these animals are entirely dependent on their camouflage skills. That's the context that makes shadowing even more essential. The trumpetfish can't simply chase down fast-moving prey. It needs to get close without being detected, because the chase phase just isn't a viable option. The entire feeding strategy depends on closing the distance before the prey bolts. The trumpetfish may also benefit from being less visible to its own predators, as well as to its prey. Hiding this way may also cut down on aggressive encounters with territorial species. The behavior, then, serves more than just hunting. It also serves defense. An animal in a shadowing position is simultaneously harder for prey to spot, less visible to predators, and less provocative to territorial fish defending sections of the reef. The choice of shield fish isn't random. The trumpetfish selects species whose coloration is compatible with its own, maximizing the visual coherence of the pairing. A mismatched color combination would draw attention. A harmonious one fuses the two animals into a silhouette that prey doesn't process as a known threat. Behavioral camouflage is, in this way, enhanced by an additional layer of functionally aesthetic judgment. The trumpetfish's shadowing behavior appears to be a useful strategy for improving its hunting success. We may see this behavior becoming more common in the future as fewer reef structures are available for them to hide in. The connection to reef degradation is telling. As corals die and the three-dimensional structure of the environment simplifies, physical hiding spots disappear. The trumpetfish appears to compensate for that loss with a behavioral strategy, substituting structural shelter with social shelter. In an earlier study, the team of scientists from the University of Cambridge found that shadowing behavior was more common in less complex habitats, such as irregular coral reefs. That correlation suggests that shadowing isn't just opportunistic behavior, but a calibrated response to the availability of environmental resources. On healthy reefs, the trumpetfish hides among coral structures. On degraded reefs, it hides among other fish. What sets this case apart from every other documented example in the animal kingdom is precisely the nature of the resource being used. Animals use tools, rocks, sticks, shells. Animals use the environment, shadows, substrate, vegetation. No other non-human animal had ever been documented using a living organism of a different species as an active concealment tool in real time during a hunting operation. The trumpetfish does it routinely, turning the presence of another living being into a calculated tactical advantage. The lantern shark, *Etmoptera spinax*, measures between 45 and 60 centimeters long and lives at depths ranging from 70 to 2,000 meters in the Atlantic Ocean and the Mediterranean. Its belly is covered in photophores, light-emitting organs, that produce continuous blue-green bioluminescence. The mechanism is counterillumination. It produces its own light on its underside to offset the silhouetted casts against the faint light coming from above, making itself virtually invisible to predators swimming below. The intensity of the light it emits is calibrated to match the ambient light. The shark adjusts its output as depth changes during vertical migrations. Beyond camouflage, photophore patterns are species-specific and likely function as a recognition system between individuals in the dark, communication by light in a sunless environment, between animals that would have no other way of visually finding each other. The most obvious solution to not being seen is to hide. The lantern shark found a different one: erase the luminous trail the body itself projects into the environment. A predator looking upward toward the surface sees dark silhouettes against a lighter background. The lantern shark eliminates that silhouette by emitting light that blends in with the surroundings. In the ocean's twilight zone, which receives minimal sunlight, bioluminescent animals can hide their silhouette from predators lurking below them by producing enough light to blend into the environment, a trick known as counterillumination. All three species analyzed showed high concentrations of photocytes on their underside, suggesting these sharks can conceal themselves from predators in exactly this way. Edmopterus spinax is capable of emitting a blue-green ventral glow, max equals 486 nanometers, through thousands of tiny photophores scattered across the ventral epidermis. The photophores consist of a cluster of photogenic cells wrapped in a pigmented sheath and covered by a lens. Pigmented cells that function like an iris are also located between the lens and the photogenic cells. This layered structure, lens, biological iris, emitting cell, allows precise regulation of how much light exits each individual photophore. Control of bioluminescence involves hormones. Melatonin and prolactin trigger light emission, while melanocyte-stimulating hormone, alpha-MSH, and adrenocorticotropic hormone, ACTH, play an inhibitory role. This hormonal regulation system is analogous to what controls skin pigmentation in other animals. The same chemical signals, repurposed to control light instead of color. This suggests the bioluminescence mechanism was built on top of already existing biological pathways. Scientists believe that nerves and hormones primarily control the light, with pigments also moving within cells as part of the process. Real-time regulation is necessary because the shark's depth changes constantly during daily vertical migrations, descending during the day, rising at night. Each change in depth alters ambient light intensity, requiring an immediate adjustment of output to maintain effective camouflage. Thanks to these organs, the luminous shark is capable of producing a particularly intense blue-green light on its ventral side, where photophore concentration is highest. Seen from below, the shark's illuminated silhouette would help it disappear by closely resembling the light coming from the surface. The 486 nanometer wavelength isn't arbitrary. It corresponds to the deep blue that dominates the mesopelagic layers after other parts of the spectrum are preferentially absorbed by the water column. Sharks use internal fertilization, so the presence of photophores on the sexual organs may facilitate mating. It could also be a way for sharks to signal that they're ready to mate, or that they're better candidates for reproduction. Bioluminescence, therefore, serves at least three simultaneous functions: camouflage against predators, intraspecific communication, and reproductive signaling. A single system handles roles that other animals solve with separate organs and separate strategies. Bioluminescence can be found in other species, the most familiar being the firefly. This ability is rare in marine organisms and even more uncommon in vertebrates, especially ones this size. That rarity in vertebrates makes the lantern shark a subject of particular interest for biomimetics researchers. Adaptive lighting systems with hormonal regulation have potential applications in display technologies and optical sensing. The mechanism these cartilaginous fish use to generate light is still unknown, since luciferin and luciferase, the molecules responsible for this ability in other animals, have not been found, nor have bacteria that could produce it. This means the underlying chemical reaction behind this shark's bioluminescence is chemically distinct from every other known light-producing system, a completely independent biochemical pathway arriving at the same physical result. The lantern shark thus inhabits a niche defined by absence of sufficient sunlight, absence of physical hiding spots, absence of any solid surface to tuck behind. In that void lit only by traces of diffuse light, the answer was to build its own light, not to see, but to disappear. The mimic octopus is remarkable for its ability to impersonate a wide variety of other marine animals. While many animals mimic their environment or other animals to avoid predation, the mimic octopus and its close relative the wonderpus are the only ones known to actively impersonate multiple animals to escape predators. Studies have revealed that this extraordinary species can successfully mimic at least 16 different animals: flounder, sea snake, squid, stingray, brittle star, lionfish, snake, eel, jawfish, blenny, mantis shrimp, ghost crab, hermit crab, jellyfish, and sand anemone. To mimic the lionfish, it spreads its arms radially, simulating venomous spines. To mimic the sea snake, it hides 6 arms in a hole and leaves 2 extended outside, undulating them with black and white bands. To mimic a flounder, it flattens its body and glides along the bottom with all arms trailing behind. It is the first animal of any kind known to alternate between multiple impersonations, a talent called dynamic mimicry. Most animals that impersonate another species practice a fixed imitation. The same appearance, always, regardless of context. An insect that looks like a leaf always looks like a leaf. The mimic octopus wasn't identified by science until the 1990s. Since then, researchers have observed that it can reproduce the shape, movements, and even behavior of more than 15 different marine species. The novelty isn't just the number of impersonations, it's the contextual choice between them. Among its most common impersonations are the lionfish, the sea snake, and the stingray. Each of these imitations is carefully selected based on the predator the octopus is trying to avoid. This implies that the animal identifies the predator at hand, retrieves from memory the most effective imitation against that specific species, and executes the transformation. 3 distinct cognitive operations in fractions of a second. Based on observations made, researchers believe the mimic octopus may decide which animal to impersonate depending on which predator is stalking it. For example, when attacked by damselfish, it takes on the shape of a banded sea snake, which is a natural predator of that fish. The octopus mimics the snake by adopting black and yellow coloring, burying 6 of its tentacles while waving the other 2 in opposite directions. The sea snake is precisely the damselfish's natural predator. The chosen imitation isn't the most generic one available, but the most specifically threatening to that particular attacker. Instead of simply camouflaging itself in the environment, it literally alters its body posture to convince other animals it's an entirely different species. The secret lies in an extraordinary combination of flexible muscles, color-changing cells, and a highly developed nervous system. Each of the octopus's arms has a high degree of autonomy to process information and react quickly to its surroundings. The physical execution of each imitation requires independent control of 8 arms, simultaneous alteration of skin color and texture patterns, and modulation of the body's overall posture. To mimic the lionfish, the arms spread radially like spines, a structure that doesn't exist on the octopus's body. To mimic the stingray, the mantle flattens and the arms widen laterally, reproducing the disc-shaped form. The mimic octopus is the first known species to impersonate other marine species. Many octopus species are extremely flexible. For example, an octopus the size of a volleyball can compress its body down to the size of a soda can. That degree of flexibility helps explain how it's able to impersonate so many different animals. Scientific observations indicate it can select the most appropriate imitation for each specific threat, suggesting an extremely sophisticated level of decision-making for an invertebrate. Cephalopods don't have a cerebral cortex like vertebrates. Intelligence is distributed. About 2/3 of an octopus's neurons are in its arms, not in its central brain. The decision about which imitation to use may therefore emerge from information processing distributed between the central nervous system and the ganglion of each arm. The mimic octopus, Thermoctopus mimicus, is unique in mimicking many animals, including toxic vertebrates. The distinction between mimicking a venomous animal versus simply mimicking a larger one points to additional sophistication. The octopus seems to identify not just the form of the most effective imitation, but the relevant property, toxicity, size, speed, that will make the predator most reluctant to attack. Dynamic mimicry, therefore, represents a layered survival strategy. First, change fast. Second, change to the right imitation. Third, maintain that imitation with enough behavioral precision to fool a predator that knows the mimicked species well. An animal without a spine, with a distributed brain and a lifespan of just a few years, solves in real time a problem of pattern recognition, contextual memory, and motor control that artificial intelligence researchers are still working to convincingly replicate. Every fall in the Caribbean, when the first storms roll in and the water temperature drops, the spiny lobster Panulirus argus abandons the shallow reefs and begins a migration to deeper waters. Not individually, in a line. The mass migration to deeper waters happens annually in Florida during late October and November, when water temperatures start to fall. The lobsters line up single file, maintained by contact between one individual's antennae and the legs of the one ahead, reducing hydrodynamic drag during the migration. Lobsters in groups of 40 or more traveling at 35 centimeters per second achieved a 65% drag reduction, a form of hydrodynamic slipstreaming, compared to lobsters traveling alone. Up to 50 lobsters can be in any given line, with each animal maintaining contact with the next using its antennae. The lobsters navigate by the direction of currents and the oscillation of waves, finding their way without vision, without GPS, and without any memory of the route, guided solely by the physics of the water around them. Drag reduction through formation travel is a principle that engineers apply in military aircraft, cycling pelotons, and high-speed trains. The animal that figured this out on its own, without theory, without controlled experiments, simply by repeating the behavior that costs the least energy. That animal is the spiny lobster. Simultaneous with the first severe fall storm, the population moves synchronously in Q formations, often both day and night. Temperature drops caused by storms statistically correlate with the onset of hyperactivity and Q formation in captive lobsters. The queuing behavior serves the migration by reducing hydrodynamic drag for each individual. The trigger is physical and measurable, a temperature drop, not a calendar date. Lobsters in groups of 40 or more traveling at 35 centimeters per second achieved 65% drag reduction, a form of hydrodynamic drafting, compared to lobsters traveling alone. The authors noted that the drag reduction implies a higher sustainable collective speed, and identified the drag reduction advantage for weaker lobsters as enabling a faster walking pace. The line doesn't just benefit the strongest individuals, it creates conditions for the weakest to keep up with the migration pace, ensuring the whole group arrives together. The initiation, posture, and alignment of a cue can be achieved entirely through tactile stimuli received by the antennae. There's no visual communication, no centralized leadership, no map. Each lobster maintains physical contact with the one ahead and adjusts its speed and direction based on the forces it feels transmitted through its antennae. The line self-organizes from simple local rules, producing a collective behavior that would reduce each individual's energy cost by 2/3. Spiny lobsters migrating in queues reduce drag by up to 65% per lobster compared to individuals migrating alone. Ducks swimming together on the water's surface reduce metabolic costs by up to 64% per duck compared to those swimming alone. The same percentage gain showing up in crustaceans and birds points to a physical principle that's independent of taxonomy. Any moving object creates a wake of lower resistance behind it, and any object that occupies that wake works less to maintain the same speed. Hydrodynamic stimuli, water currents and wave oscillations, serve as orientation guides under certain conditions. Navigation without visual reference works because wave oscillation has a predominant direction that doesn't change randomly. The lobsters read water flow with mechanical receptors distributed across their bodies and use that information as a physical compass. Not magnetic, not visual, but purely mechanical. Females move to deeper waters for spawning, and mass migrations occur in the fall, when the animals, in single-file lines of up to 50 individuals, move in a certain direction during the day, each animal maintaining body contact with the next through its antennae. The continuous physical contact means the line functions as a coupled mechanical system. Disturbances at the front propagate backward like waves, and each animal responds to those waves by adjusting its pace. Lines of migrating lobsters cross long stretches of open sandy substrate during daylight hours near Bimini, Bahamas. All cues orient southward, parallel to the edge of the Great Bahama Bank, following late fall stormy weather episodes. The consistently southward orientation along the continental shelf edge reveals that the navigation isn't random. There's a directional vector maintained throughout the entire migration, likely derived from the combination of current flow and wave oscillation. What the spiny lobster demonstrates is that sophisticated collective behavior doesn't require a sophisticated nervous system. It doesn't require individual recognition, symbolic communication, or route memory. It only requires that each individual follow one simple rule: maintain tactile contact with the one ahead, and that this rule, repeated by dozens of animals simultaneously, generates an emergent pattern that no individual alone could produce or even begin to understand. Cuvier's beaked whales hold the records for both the deepest dive and the longest dive ever documented among mammals. Recorded dives have reached depths of 2,992 meters and durations of 222 minutes. At 2,992 meters, the pressure is approximately 300 atmospheres, the equivalent of what destroyed OceanGate's Titan submersible at 3,800 meters in milliseconds. The Cuvier's beaked whale survives because it has no rigid air cavities in its body. The lungs collapse during the dive, and oxygen is stored directly in the hemoglobin and myoglobin of the blood and muscles. Its muscles are loaded with myoglobin, the protein that binds oxygen and gives whale muscle its characteristic dark red or near black color. Calculations suggested these relatively small mammals should only be able to stay submerged for about 33 minutes before running out of oxygen. But experience showed they were capable of diving for far longer. The longest recorded dive lasted 222 minutes, 12 minutes longer than the movie Titanic. Any submarine descending to 3 kilometers needs a reinforced hull, active pressurization systems, and careful engineering to avoid being crushed. The Cuvier's beaked whale reaches the same depth with none of those structures, because it built the solution inside its own body. It holds the records for the deepest and longest dives ever recorded among whales at 2,992 meters and 222 minutes, respectively, though the frequency and reasons for these extraordinary dives are not fully understood. The numbers alone are impressive. What makes them even more remarkable is the size of the animal pulling them off. The Cuvier's beaked whale measures between 5 and 7 meters and weighs approximately 2 tons. Not the largest whale, and not the one you'd intuitively expect to be the most capable of extreme diving. One of the most important strategies is lung collapse. When diving, the chest and lungs of cetaceans collapse, expelling most of the air. This prevents excessive nitrogen from being absorbed into the blood under high pressure, avoiding decompression sickness. The oxygen needed for the dive isn't stored in the lungs. It's stored in the blood and muscles. That's the core mechanism. Rigid lungs full of air under 300 atmospheres of pressure would be crushed. Flexible lungs that collapse simply yield to the pressure without rupturing. And the oxygen has already been transferred to other structures before the descent even begins. Myoglobin, or muscle protein, stores oxygen and is present in nearly all vertebrates. This protein gives whale and dolphin meat its dark color. It stockpiles oxygen, releasing it to cells when needed. The muscles of deep-diving whales contain concentrations of myoglobin orders of magnitude higher than those found in equivalent land mammals. The more myoglobin, the more oxygen available in muscle tissue, independent of whatever oxygen the blood is carrying. Marine mammals have a higher blood volume and more hemoglobin than land mammals of comparable size. Myoglobin occurs in muscle tissue. Whale muscle has a higher concentration of myoglobin than the muscle of land mammals. The combined effect of increased hemoglobin in the blood and elevated myoglobin in the muscles means the total oxygen supply available during a dive far exceeds what the lungs could store on their own. Underwater, whales can slow their heart rate and redirect oxygenated blood to the areas that need it most, such as the brain, heart, and muscles. This active redistribution is controlled by the autonomic nervous system. It doesn't require any conscious decision. The body simply executes the dive protocol, reduces blood flow to hypoxia-tolerant tissues, prioritizes the heart and brain, and drops the overall heart rate to burn less oxygen per unit of time. In 2020, a Cuvier's beaked whale set a new dive record, staying underwater for 3 hours and 42 minutes. That data point, however, was excluded from the study's official results because it occurred following exposure to sonar used by researchers, which may have affected the whale's behavior. Mid-frequency military sonar alters the behavior of these whales in ways that are still poorly understood. And it is also the primary factor associated with mass strandings of the species, making it a subject of both scientific and conservation concern. The blood of these whales likely has high concentrations of the proteins myoglobin and hemoglobin, allowing them to store far more oxygen than other mammals. Researchers calculated that the observed dives far exceeded what aerobic consumption models had predicted. The gap between the predicted and the actual was so large that it suggested some additional metabolic mechanism, not yet fully understood, contributes to the species' performance. One characteristic that allows whales to remain submerged for extended periods is the high concentration of myoglobin in their muscles. The near-black color of the musculature is visible in stranded specimens, an immediate external indicator that tissue was optimized not for strength or speed, but for oxygen storage under extreme pressure and with zero access to air. The Cuvier's beaked whale dives to hunt squid and deep-sea fish that live where the pressure would destroy any submersible not specifically engineered for it. It surfaces, breathes for a few minutes, and goes back down. Each dive is an excursion into an environment that remains inaccessible to humans without specialized technology, carried out by a 2-ton mammal that breathes air and carries all the necessary equipment for the mission inside its own body. The adult Nemicthes scolopaceus can reach 1.5 meters in length, while weighing only a few grams. It has more vertebrae than any other known animal, around 750. Its anus migrated to its throat during development. Its upper and lower jaws curve in opposite directions and never close completely, a structure perfectly suited for snagging shrimp by their legs mid-motion. But before it becomes that animal, it is something else entirely. The larvae are transparent, leaf-shaped, and inhabit the upper 200 meters of the water column, primarily the top 150 meters, where they carry out daily vertical migration, feeding in these larvae centers on marine snow. Their larvae are leaf-shaped and actually get smaller before transforming into adults. The leptocephalus shrinks during metamorphosis. It loses length, gains density, and emerges as a completely different animal from what it was during months or years of drifting. From the mantis shrimp that strikes with 1,500 newtons to the leptocephalus that shrinks before growing, the ocean has animals that break expectations at every scale, from millimeters to kilometers of depth. Every known metamorphosis involves growth. A larva accumulates resources, increases in mass and complexity, and emerges as a larger adult. The snipe eel reverses that sequence at a critical point. Before becoming an adult, it shrinks. Length decreases, the laminar structure dissolves, and what emerges is a completely different animal. Not a larger adult, but one reorganized from less material. Their larvae are leaf-shaped and actually get even smaller before transforming into adults. The leptocephalus, the name given to this transparent, flattened larva, can reach 45 centimeters in length while still in its leaf-shaped phase. The metamorphosis that follows doesn't just change its form. It literally reduces the organism's total size before reorganizing it into the adult body plan. A leptocephalus larva is typically elongated, laterally compressed, transparent, and gelatinous. In the case of the snipe eel Nemicthes scolopaceus, larvae can reach 45 centimeters in length. The transparency isn't just an optical coincidence, it's a strategy. An organism that's invisible in the water column is difficult to detect by both predators and prey. The gelatin filling the body functions like structured water. It provides shape without a high metabolic cost. In the predatory world of the twilight zone, creatures tend to shed every part of the body that doesn't serve them. Over time, the snipe eel took that minimalism to an extreme, reducing its body to little more than a thread with a head at one end. In the case of N. scolopaceus, the only place the anus could fit is at the throat, which is slightly wider. Imagine a body so narrow that its internal proportions had to be completely recalibrated. Organs that would normally sit in the abdominal region simply have no room there. The anatomical solution was to reposition them. It has more vertebrae in its spine than any other known animal, around 750. Its body is so slender that its length is 75 times greater than its width. It weighs just a few grams despite its considerable body length. For comparison, humans have 33 vertebrae. The snipe eel's spine, distributed across 1.5 meters of length at thread-like thickness, functions as a support skeleton for an organism that is essentially length without volume. The upper and lower jaws curve in opposite directions and never close completely. In the period following metamorphosis and the onset of sexual maturity, they have long, slender jaws packed with very fine, densely spaced teeth. Sexually mature males lose their teeth and undergo a drastic change in appearance. This loss of teeth at sexual maturity is so extreme that mature males were cataloged for decades as entirely separate species. Only molecular analysis confirmed they were the same animal at a different stage. During metamorphosis, elopomorphs transform into a cylindrical shape as there is a reduction in both the length and depth of the body, along with loss of teeth and thickening and pigmentation of the skin. In the specific case of the snipe eel, this transformation means shedding the transparent leaf form and taking on the dark adult filament. Two organisms so morphologically distinct that, without genetic analysis, they would appear completely unrelated. Adults can be found from the surface down to 4,000 meters in the North Atlantic and carry out daily vertical migrations. The same animal born as a transparent leaf drifting in the first 150 meters of the water column ends its life making vertical round trips of several kilometers, every single day, in the dark, hunting shrimp with jaws that never fully close. The snipe eel closes, with a kind of symbolic precision, a catalog of animals that defies expectations at every possible scale. From the mantis shrimp that strikes at bullet speed to the snipe eel that shrinks before it grows, the ocean continuously generates solutions to problems in physics, chemistry, and biology that no engineer commissioned and no laboratory anticipated. Each species is a different answer to the same question. How do you survive in an environment that wasn't designed to be habitable, and do it efficiently enough to thrive for millions of years, while the ocean changes around you without warning and without compromise?